Chlorophyll fluorescence is a powerful tool for assessing the function of photosynthesis in plants. To understand the fluorescence signal, we will review the basics of photosynthesis. Only plants, algae, and cyanobacteria can perform photosynthesis. It is the basis for almost all life on Earth. Photosynthesis produces oxygen, which is necessary for many living organisms. The green, spherical organelles within plant cells are called chloroplasts. Photosynthesis takes place within these chloroplasts. Photosynthesis is spatially organized. Within the chloroplasts, the thylakoid membrane hosts the light reactions, where water is oxidized. Electrons are transferred through the photosynthetic electron transport chain, and energy-rich molecules are generated. Inside, there are membranes that form a network of flattened sacs. These are called thylakoids and are the site of the light reaction. They contain special protein complexes that contain pigments or clusters of iron, copper, or manganese to enable them to absorb light energy or transfer electrons along a chain of different redox reactions. Photosynthesis consists of two processes, the light reactions and carbon fixation. The light reactions take place in the thylakoid membrane of chloroplasts, where light energy drives water oxidation and oxygen is produced. Electrons are transported through the photosynthetic electron transport chain, facilitating the production of ADP and NADPH, two energy-rich molecules. These molecules are then used in carbon fixation to bind carbon dioxide and synthesize sugars. The efficiency of the light reactions can be assessed using chlorophyll fluorescence measurements. Initially, the light energy is absorbed by chlorophyll in the antenna complexes and directed to the reaction centers, where it excites an electron to such an extent that it leaves its original position and moves to an acceptor molecule. This process is called charge separation. The reaction center now has a positive charge which will draw a replacement electron into it. There are two types of photosystems, Photosystem I and Photosystem II. At Photosystem II, the replacement electron comes from its water-splitting complex. The water-splitting complex splits water molecules into oxygen, protons, and electrons. The protons are released into the thylakoids. Photosystem I receives its electrons from plastocyanin, which is at the end of a well-organized electron transport chain, starting at Photosystem II. As the electrons move through the chain, additional protons are pumped into the thylakoid lumen, creating a proton motive force across the thylakoid membrane. This gradient is used by the ATP synthase complexes to produce ATP. The electrons generated by Photosystem I are used to reduce NADP to NADPH. ATP and NADPH are energy-rich and power the carbon fixation. The blue curve shows the absorption spectrum of chlorophyll A. It absorbs light most strongly in the blue region, around 430 nanometers, and the red region, around 660 to 680 nanometers. Absorption is much weaker in the green range. This is why leaves appear green. The red curve shows the fluorescence emission of chlorophyll A. After excitation, a small amount of the absorbed energy is re-emitted as light at a longer wavelength, with the main peak occurring around 685 to 690 nanometers. These basic features are important for chlorophyll fluorescence because the inner antenna of Photosystem II is the main source of this fluorescence, which is emitted as red and far-red light. When a chlorophyll molecule absorbs light, its electrons gain energy. This energy can be used in three ways. It can be passed to other pigments until it reaches the reaction center and drives photosynthesis. It can be released as heat, or it can be emitted as weak fluorescent light. Changes in how efficiently energy is used for photosynthesis or released as heat show up as changes in fluorescence. This is why chlorophyll fluorescence can be used as a non-invasive way to study photosynthetic activity. Chlorophyll fluorescence can be measured using modern chlorophyll fluorometers. Walz offers PAM chlorophyll fluorometers for a wide range of applications. Instruments are available for laboratory use, monitoring underwater applications, and imaging systems. The system supports single-point measurements, suspension measurements, and spatially resolved imaging of leaves and whole plants. Measurements are based on the PAM technique, enabling sensitive and quantitative analysis of photosynthetic activity. The abbreviation PAM stands for Pulse Amplitude Modulation. In this method, fluorescence is detected exclusively from light that is modulated at a defined frequency. The modulated illumination is called the measuring light. When this light interacts with a photosynthetic sample, it induces a correspondingly modulated fluorescence signal. The instrument directly extracts and indicates the fluorescence amplitude, and therefore selectively detects only the fluorescence that is induced by the modulated measuring light. Fluorescence caused by continuous illumination, whether ambient sunlight or actinic light applied during an experiment, is not detected directly by the system. Instead, the detector locks onto the specific frequency of the modulated measuring pulses and extracts only the corresponding fluorescence component. As a result, the PAM signal reflects only changes in the photochemical state of Photosystem II, since they influence the amplitude of the modulated fluorescence. This selectivity allows assessment of Photosystem II under a wide range of ambient light conditions, including full sunlight. To assess PS2 activity, the saturation pulse method is used. A very weak modulated measuring light is applied continuously. It is strong enough to probe chlorophyll fluorescence, but too weak to drive photosynthesis in any relevant way. Under these conditions, PS2 reaction centers remain open. A short, intense saturation pulse temporarily closes all Photosystem II reaction centers by fully reducing the primary electron acceptor QA and filling the electron transport chain with electrons. As a consequence, charge separation becomes very slow and fluorescence rises to its maximum level. Because electron transport is temporarily blocked, excitation energy can no longer be used photochemically and is instead released as fluorescence. From these fluorescence levels, key parameters can be calculated that describe PSI efficiency and energy dissipation. In practice, fluorescence measurements are typically described using several standard fluorescence levels. These levels can be used to evaluate a wide range of parameters, for example, the efficiency of photochemistry or the degree of protection against excessive light. We will walk you through a typical PAM saturation pulse experiment step by step. The experiment begins with an induction phase, followed by a recovery phase. First, the sample is dark adapted to ensure that all PS2 reaction centers are fully open. Open means that the electron acceptors of PS2 are oxidized and can take electrons from the reaction center. Also, the enzymes of the carbon fixation shall become inactive. Any protection mechanisms against excessive light that quench the fluorescence level shall relax. This typically takes 10 to 30 minutes or longer if necessary. This dark period allows us to start from a well-defined baseline. The weak modulated measuring light is switched on to monitor this baseline. This value, F0, represents the fluorescence emitted by PS2 when its primary acceptor QA is oxidized and all PS2 reaction centers are open. It provides the reference point for the subsequent saturation pulse step. Next, we apply a short, intense saturation pulse. The pulse is strong enough to transiently close all Photosystem II reaction centers by fully reducing the primary acceptor, QA, and filling the complete electron transport chain with electrons. As a result, charge separation becomes very slow and fluorescence rises to its maximum level. Because electron transport is temporarily blocked, excitation energy can no longer be used photochemically and is instead released as fluorescence. As soon as the pulse ends, fluorescence rapidly drops back toward the baseline as electron transport resumes and the reaction centers reopen. From this initial saturation pulse, we obtain the minimal fluorescence level F0 and the maximal fluorescence level Fm. Together, these two values define the fluorescence range of the dark adapted state. The difference between these two values is defined as the variable fluorescence FV. The ratio FV over FM is commonly used as a simple indicator of the maximum quantum efficiency of Photosystem II in a dark-adapted sample. For many plant species, values around 0.79 to 0.84 are considered typical for healthy, unstressed leaves. Lower FV over FM values usually indicate photoinhibition or other forms of physiological stress affecting Photosystem II. To assess how the plant adapts to light, we switch on actinic light at a defined intensity to drive photosynthesis. During this actinic period, we repeatedly apply saturation pulses. This measurement allows us to track changes in fluorescence and quantify the different quenching processes that develop in the plant over time. In this light-adapted state, the maximal fluorescence level reached during each saturation pulse is defined as FM', while the fluorescence level immediately before the pulse is denoted as F. Each pulse therefore provides a value for FM', and together with a corresponding F value, the effective quantum yield of Photosystem II can be calculated. The shape of the fluorescence trace reflects the plant's acclimation to the applied light regime. Under sustained illumination, not all excitation energy can be utilized for photochemistry, and therefore protective mechanisms are activated to dissipate excess energy safely as heat. This regulated energy dissipation is referred to as non-photochemical quenching (NPQ). During the induction phase, NPQ typically increases as the sample adapts to light, and the fluorescence signal eventually approaches a steady state once a new balance between photochemistry and energy dissipation has been established. NPQ is calculated by comparing the maximal fluorescence measured in the dark, FM, with the maximal fluorescence measured in the light, FM'. After the actinic light is switched off, the sample gradually recovers in the dark as non-photochemical quenching relaxes and the photosynthetic apparatus returns toward its pre-illumination state. This unquenching can be followed over time by applying additional saturation pulses, allowing the kinetics of recovery to be quantified. Taken together, PAM chlorophyll fluorescence measurements are a versatile tool for studying photosynthesis in both the laboratory and the field. Please visit our website for more information and explore our other online tutorials.